So microorganisms act as a catalyst in any fermentation where a product is obtained. When the culture is removed from the fermenter or the bioreactor, the catalyst is either destroyed by subsequent downstream processing or discarded. It is possible to harvest cells and reuse them, but there are a number of practical problems. That is, enzyme activity is lost unless there is a further period of cell growth, and contamination, of course, is the major problem, is what until we saw. And one solution is to immobilize the microorganisms, even, of course, the appropriate enzyme. So to make it reuse, or in order to avoid its contamination, or in order to make its functional stable, you can immobilize what is called as enzyme or the microorganisms that are used as catalyst in an fermenter reaction. And a very important 2-mark definition. As such, enzyme immobilization, if you see, is a very important 5-mark question. In case if it is specifically asked, it will be asked in what is called as 2-mark. That is its definition. Regarding enzyme immobilization, the definition: immobilization of enzymes or cells refers to the technique confining to anchoring the enzymes or cell in or on inert support for their stability and functional reuse. So, either inert material kuda or enzyme combine panni, what is immobilization is fixing it. Adhere panni vechiruvanga. It will not be able to move. That is why it is called as immobilize. So in case if you are immobilizing an inert support an enzyme or a cell, you call the phenomenon as immobilization. I repeat, immobilization of enzymes refers to the technique confining to anchoring the enzymes or cells in or on inert support for their stability and functional reuse. Shall we go on to the next slide?
Yes, ma'am.
Okay. So what are the advantages of immobilized enzyme? Why are we doing the technique of immobilization, and what are its advantages? The first advantage is they are stable. The enzyme or cells that are immobilized are stable and they are more efficient in their function. Can be used or reused again and again. So you can reuse the enzyme without discarding it again and again. Products are enzyme free. So during a fermentation reaction, when microorganisms or enzyme are used as catalyst, the final recovery that you do, the recovery process should be free from enzyme or should be free from the catalyst. So when you obtain a product using an immobilized enzyme, the products are enzyme free. And it is ideal for multi-enzyme reaction system. Imagine a situation, you have a bioreactor or a fermenter. You are supposed to use more than one enzyme as a catalyst. You can use immobilized cell or immobilized enzyme. So it is ideal for multi-enzyme reaction system. The control of enzyme function is very, very easy because it is immobilized onto a surface. So to control its function is very, very easy. Suitable for industrial and medical use. It is of course suitable for both industrial and medical use. Minimize the effluent disposal problem. So when you go in for disposal of effluent is waste, dispose pannumbodhu you will have minimum effluent disposal problem when you use immobilized enzyme. I repeat, advantages of immobilized enzyme: they are stable and more efficient in function. It can be used again and again. Products are enzyme free, ideal for multi-enzyme reaction system. Control of enzyme function is easy. It is suitable for industrial and medical use, and of course minimize effluent disposal problem. So when you have an advantage for a procedure, you will equally have disadvantage as well. So comparatively, advantages are more than disadvantage when you immobilize a cell or enzyme. Two disadvantage we find when we immobilize. If you see, the possibility of loss of biological activity of an enzyme during immobilization or while it is in use. And of course, the other disadvantage we have is it requires sophisticated equipment to bring about the process of immobilization. So two disadvantage we have is the possibility of loss of biological activity of an enzyme during immobilization or while it is in use, and it is an expensive affair often requiring sophisticated equipment. I repeat the advantage and disadvantage of immobilization. Advantage of immobilized enzyme: they are stable and more efficient in function, can be used again and again. Products are enzyme free. It is ideal for multi-enzyme reaction system. Control of enzyme function is absolutely easy, suitable for industrial and medical use, and of course it minimizes effluent disposal problem. While disadvantage include the possibility of loss of biological activity of an enzyme during immobilization or while it is in use, and of course it is an expensive affair, often requiring sophisticated equipment. Shall we go on to the next slide?
Yes, ma'am.
What are the choices? You have various methods of immobilization, and what and how do you choose the method? What are the choice that will make for an immobilization technique? So the choice depends on factors such as the operational stability of the immobilized catalyst, a very important 2-mark question again. The operational stability of the immobilized catalyst, the cost of immobilized catalyst, the activity, and of course the yield of immobilized catalyst, the regenerability of the catalyst, and of course the cost of the appropriate reactor configuration. So when you go in for immobilized catalyst, you should see the process should not reduce the stability, number one. Number two is how far is the cost of immobilized catalyst. Of course, if you see, imagine your product is a very cheap product, and if you're going to use the immobilized method that is very, very costly, it is of course going to be waste. So you have to check out whether the cost of immobilized catalyst will suit the product, number one. And of course, whether the immobilized catalyst will give you a very high and active yield, that is another one. Can the immobilized catalyst have the function of regenerability, that is whether it can regenerate for its function, is one more thing. And what is the appropriate cost of the reactor configuration? You're going to produce immobilized cellular enzyme, and what will be the appropriate configuration of the reactor that you're going to use? I repeat again, choice of immobilization methods, it depends on the factors such as a very important 2-mark question again. List out the factors involved in the choice of immobilization method: the operational stability of the immobilized catalyst, the cost of immobilized catalyst, the activity and yield of immobilized catalyst, the regenerability of the catalyst, and of course the cost of the appropriate reactor configuration. So what are the various method or different methods of immobilization? And of course, the various method that you will be studying is equally applicable for both the cells and enzymes. The methods of immobilization available are equally applicable to cells and enzymes. The first method you have is physical binding. The second method you will have is cross-linking. And the third one we have is entrapment. Cross-linking is further divided into covalent bonding and copolymerization. Under entrapment, you will study another heading, micro encapsulation. I repeat, immobilization, physical binding, cross-linking, and entrapment. Cross-linking, you have covalent bonding, copolymerization. Under entrapment, you will have micro encapsulation. So all methods will not be used for immobilizing all the enzymes or cell. Based on its function and based on what for you're going to use the cell and enzyme, whether the cell and enzyme may remain stable by this method, you will have the choice of one among these different methods that will be equally applicable both for cells and enzymes. Shall we pass?
Yes, ma'am.
Okay. The first one, physical binding. Physical binding is also referred to as adsorption. If you're going to look into the right picture you have, M is the matrix, inert matrix. E is the enzyme. Just fixing by physical binding to an inert matrix is called as adsorption. The physical binding is the oldest immobilization technique, of course, but least satisfactory. Cells or enzymes are mixed with adsorbent. The M column is called as adsorbent. So adsorbent and then packed in an column. Adsorption of enzyme molecule or inert support involves weak forces such as van der Waals forces and hydrogen bonding. So in the procedure, like physical binding la ஏற்படக்கூடிய bonds or chemical force என்னன்னு பார்த்தோம் அப்படின்னா van der Waal force and hydrogen bond. இது ரெண்டுமே weak forces. So எவ்வளோக்கு எவ்வளோ ஈஸியா பிசிக்கலா பைண்ட் ஆகுமோ அவ்வளோ அதை பிரிச்சிடலாம், ஈக்குவலாவும் பிரிக்கலாம். So the adsorbed enzyme can be easily removed by minor changes in pH, ionic strength or temperature. What are the adsorbents used if you see is என்னெல்லாம் inert material use பண்ணுவாங்க for enzyme fixing is alumina, silica gel, starch and of course CMC, carboxymethyl cellulose. I repeat, physical binding, the oldest immobilization technique but least satisfactory. Cells, enzymes are mixed with adsorbent and then packed in a column. Adsorption of enzyme molecule on inert support involves weak forces such as van der Waals forces and hydrogen bond. So the adsorbed enzyme can be easily removed by minor changes in pH, ionic strength or temperature. And the adsorbents used include alumina, silica gel, starch and carboxymethyl cellulose. Shall we go?
Yes, ma'am.
Okay. The next procedure you have is cross-linking. So here if you see, the strongest of the bond will act, the covalent bond. Enzymes or microbial cells can be immobilized by creating covalent bonds between the chemical group of enzymes and the chemical group of the support. The M is the matrix and E is the enzyme. The bond that acts here is the strongest of the bond, the covalent bond. So if you look into the right side picture, you have covalent binding and copolymerization. Cross-linking is carried out with bi or multifunctional reagents such as glutaraldehyde or toluene diisocyanate. So the chemicals that are used to bring about immobilization are multifunctional reagents, or which includes glutaraldehyde or toluene diisocyanate. One biggest disadvantage if you have with this method is because you're using chemicals to bring about bond. Although good retention of cells or enzymes is obtained, you will have extensive loss of enzyme activity. So although good retention of cells and enzymes is obtained with this method, it can be accompanied by extensive loss of enzyme activity. I repeat, enzymes, microbial cells can be immobilized by creating covalent bonds between the chemical group of enzymes and the chemical group of support. Cross-linking is carried out with bi or multifunctional reagents such as glutaraldehyde or toluene diisocyanate. Although good retention of cells and enzymes is obtained with this method, it can be accompanied by extensive loss of enzyme activity. Shall we pass?
Yes, ma'am.
Okay. The next method we have is the entrapment, and of course the extensively used method. The most extensively used method of cell immobilization involves entrapment in a polymer or a gel matrix. The size of matrix pore is such that the enzyme is retained while the substrate and product molecules passes through it. Matrices, what are the different types of matrices used for entrapment if you see is collagen, gelatin, agar, alginate, polyacrylamide, cellulose triacetate and polystyrene. I repeat, matrices which have been employed include collagen, gelatin, agar, alginate, polyacrylamide, cellulose triacetate and polystyrene. If you look into the right side diagram, two different forces play a very important role here. Just a non-covalent entrapment, just entrapping in without covalent bonding, or you can covalently bind it with the help of entrapment using matrices. So I repeat, the most extensively used method of cell immobilization involves entrapment in a polymer or gel matrix. The size of matrix pores is such that the enzyme is retained while the substrate and product molecule pass through. Matrices which have been employed includes collagen, gelatin, agar, alginate, polyacrylamide, cellulose triacetate and polystyrene. And one of the other method under entrapment if you see is micro encapsulation, which is again a type of entrapment method. It is referring to the process of spherical particle formation wherein a liquid or suspension is enclosed in a semi-permeable membrane. So the enzyme will be fixed within the semi-permeable membrane. If you look into the right side diagram, bottom one, you will see the semi-permeable membrane where you find membrane confinement. I repeat, microencapsulation is a type of entrapment. It refers to the process of spherical particle formation wherein a liquid or suspension is enclosed in a semi-permeable membrane. Shall we go on to the next slide?
Yes, ma'am.
Yes, ma'am.
So you have a selected list of important immobilized enzyme, and of course their industrial applications. So on the left column you see immobilized enzyme, on the right side column you see what is called as its applications. I will read out, just listen. Glucose isomerase, it is used for the production of high fructose syrup from glucose. Amylase, production of glucose from starch. Invertase, splitting of sucrose to glucose and fructose. Beta galactosidase, splitting of lactose to glucose and galactose. Penicillin acylase is used for the commercial production of semi-synthetic penicillins. Aspartase, production of aspartic acid from fumaric acid. Fumarase, synthesis of malic acid from fumaric acid. Histidine ammonia lyase, production of uroconic acid from histidine. Ribonuclease, synthesis of nucleotides from RNA, and of course nitrilase, production of acrylamide from acrylonitrile. So left side if you see, they are all immobilized enzyme. In the immobilized enzymes எல்லாம் வச்சு என்னென்ன apply பண்ண முடியும், என்னென்ன produce பண்ண முடியும்ன்றது, it is right side column. So this column is a list of important immobilized enzyme and their industrial application. The next one will be the selected list of immobilized cell that brings out one or two enzyme reaction in industrial application. இதுக்கு முன்னாடி பார்த்த slide if you see is the immobilized enzyme. In the slide பாத்தீங்கன்னா microorganism as a whole is immobilized and they are called as microbial catalyst அப்படின்னு சொல்லுவாங்க. You have two columns again. The left side if you see is the immobilized microorganism which is microbial catalyst. On the right hand side you have its application. I will go ahead with it. Escherichia coli for the synthesis of L-aspartic acid from fumaric acid ammonia. Escherichia coli for the production of L-tryptophan from indole and serine. Pseudomonas species for the production of L-serine from glycine and methanol. Saccharomyces cerevisiae for hydrolysis of sucrose. Saccharomyces species for the large-scale production of alcohol. Zymomonas mobilis for the synthesis of sorbitol and gluconic acid from glucose and fructose. Anthrobacter simplex for the synthesis of prednisolone from hydrocortisone. Pseudomonas chlorophysis for the production of acrylamide from acrylonitrile, and Humicola species for the conversion of rifamycin B to rifamycin. Yes, these are not the only list of immobilized microorganisms and its application. We have various organisms that are still immobilized, among which a few example is cited onto your slide. Similarly, a few example of immobilized enzymes are cited onto your slide. So you have many enzymes and many microorganisms that are immobilized with its various function. Shall we go?
Yes, ma'am.
Okay.